Hi biology, this is Miss Satterley. I'm going to go over dihybrid cross squares. So we already learned how to do a monohybrid cross. A mono means one, so it's a cross between just one trait. So that's either purple flowers, white flowers, yellow seed, green seed, and so on, if we're talking about our different pea plant traits. Now we're going to talk about a dihybrid cross. So a dihybrid cross is when you're crossing two traits. So di means two. So crossing of two traits. So yesterday I posted an assignment about practicing creating dihybrid cross gametes. So when you have a Punnett square, so we've done a Punnett square with only 4 squares, and now we're upping it to 16. On the assignment, I explained why we use 16 boxes. So you have 4 combinations that you can end up with. So 4 times 4 is 16. Compared to a monohybrid cross, you only have 2 combinations, and 2 times 2 is 4. Make sure you've done that and you understand how to do that, because knowing how to do gamete formation for dihybrid crosses is very important for being able to do them. I'm going to do a cross between 2 traits that the parents are both heterozygous for both traits. I'm going to use colors as well as I go through this, just so we can keep track of what we're doing. So I'm going to use the shape of a pea seed and the color of a pea seed. Again, the shape that we have for a pea seed can either be round or wrinkled. You need to remember your pea plant traits. Round is our dominant, so we're going to use an R for that. And then color is going to be our next one. Pea seeds can either be yellow or green. Yellow is our dominant color, so we're going to use a Y. So how I'm going to do this is I'm actually going to use one color for my dominant letters and another color for my recessive letters. So not the 2 traits themselves, but capital versus lowercase, just so I can see them a little better when doing these crosses. So we're going to cross 2 heterozygotes. So first, hetero means different. So my first trait is going to be big R, little r. And my second trait is my seed color, which is going to be big Y, little y. Since both of my parents, I'm crossing both my parents here. They both need to be heterozygotes. I'm going to write both of them down. Okay, so far so good. So as we've been doing, my first parent gametes are going to go across the top. My second parent gametes are going to go to the side. So the assignment that's posted, that's due today, you're practicing how to make these gametes. You're going to use the FOIL method. I tried to make sure I explained what that meant. So FOIL is when you take first, which is the first letter of each pair. So my first gamete, and I'm going to go ahead and write them across my dihybrid boxes here. My first gamete is big R, big Y. That's first. Next is outside, which means I take my 2 outside letters, this one and this one. So I have big R with little y. Next, I stands for inside. That's my 2 inside letters. Little r, big Y. My last one is last, which would be the last letter of each combination. Little r, little y. Okay, well, Miss Satterley, I thought capital always goes before lowercase. Yes, that's true, but we have a couple rules we need to follow. The first one we're going to follow is ABC order. So when we're writing traits, ABC order is going to take place over capital before lowercase. So again, our second rule is big before little. Big before little is going to be when you have the same letter, so 2 R's, and then ABC order is going to be the other trait that we're dealing with. So that's why we have ABC order here, a little r with a y. Also, we want to have some consistency, so my r goes first in all of them, and my y goes second for all of them. Since I'm crossing the 2 parents that are the same, my gametes are the same. So I'm going to go ahead and write my gametes down the side. Okay, so now I need to fill it in. On Google Classroom, I have this blank dihybrid cross square that's posted that if you have access to a printer, you can print it out and practice your dihybrid cross, because I want you guys to do this so you understand how to figure out a dihybrid cross. If you're in person, we did this in class. And if you've been absent this week or you're a remote learner, make sure you watch this video. And again, if you have any questions, please let me know, and I will do what I can to explain everything in more detail. So how I fill this in, I do it the same way I do a monohybrid cross, except now I have 4 letters instead of 2 letters, and I need to follow my rules. ABC order, big before little. So I'm going to do my R's first. I have 2 big R's and 2 big Y's. That's my first offspring. Okay, so I'm gonna do across the top here. This is another reason why I have the 2 separate colors, just so I don't get confused if it's lowercase or capital letter. So I have 2 big R's, 1 big Y, big before little, and lowercase y. 1 big R over here, little r, big Y, and big Y. My last one I have big R, big Y, and little y. So that's my first one. I'm going to keep going. Big R, big Y, little y. Big R, little r, 2 big Y's. And in case you're wondering where I'm getting these from, the same way we do a regular Punnett square, I bring these 2 down and these 2 over and just put them together. This one down here, big R, big Y, little y. Keep going. We have 2 big R's here and 2 little Y's, sorry. Big R, little y, big Y, little y. I'm going to do this one. You'll notice that a pattern emerges. All of these in this row are all the same. They're all heterozygotes. Okay, big R, little y, little y, little r, big Y, big Y. Okay. And I'll just do my last boxes really quick so we can go on to our next part of what to do once we get our Punnett square filled out. So here it is. Our large Punnett square, we're using the same concepts from monohybrid crosses for the dihybrid cross. Okay, so what we need to do now is we need to find out our phenotypic ratio. So that's what we're going to do next. I'm going to move this up a little. So we want to figure out our phenotypic ratio. Again, phenotype is physical appearance, so what it looks like. So I have to look at the top here. Here are my different phenotypes. So we have to put these 2 traits into 4 combinations. The first thing I want to do, and I'm going to use here is I'm going to use 4 different colors for my traits. And that's going to come into play when I go back to the squares. So first one, round is the dominant. So a round and yellow seed. That's my first one, round and yellow seed. And I'll come back to that in a second. Next, I'm going to stick with round is my dominant trait, and now I need green seed. Next one that I want to do is, instead of round, I've already used round twice, so I need to do wrinkled. So we're going to have a wrinkled and yellow seed. So I have one recessive and one dominant, just like this one. One dominant, one recessive. And then my last one is going to be recessive for both traits, a wrinkled and green seed. All right, good. So now what I need to do is go up to my square here, and I'm looking at my offspring. I need to find all of the ones that are both round and yellow. Well, I know that round will have at least one big R, and this could be either another big R or little r, the second letter. And I also know that I need to have at least one big Y, and the second letter again can be either a big or little. So this is what I'm looking for, one big R and one big Y. So I come up here. Okay, here's a big R, here's a big Y, which this is a round and yellow seed. I'm going to circle this in the color that matches my phenotype as an indication. One big R, one big Y. Big, big. There it is again. Well, I know if those are all the same genotype, they must be the same phenotype. So I'll go ahead and circle those. And we have a big R and big Y. Big R, big Y. Let's check the other ones. Big R, little y. Nope. Big R, little y. Nope. Little big. No. Okay, so there are my round and yellow seeds. I need to make a ratio here. I'm going to keep track of how many round and yellow seeds. If I count them, they have 9. 1, 2, 3, 4, 5, 6, 7, 8, and 9. So now I need to do my next color. I'm looking for round and green seeds. Well, I know round is always a big R in it. So I'm looking for either 2 big R's or one that has a recessive for the next one. Green seed. Well, green is different from yellow, so I know that's a recessive, which means it only can be 2 little y's. So let's look at our back up here. So big R, 2 little y's. I'm going to circle that. Big R, 2 little y's. There's no big R's on any of those. Big R, 2 little y's. Circle. Do the same thing. I'm going to count them. 1, 2, and 3. So I have 3. Okay, wrinkled and yellow seed. Wrinkled is a recessive, so that's going to be 2 little r's. Yellow, again, we're looking for a big Y, and then either a big or a small with the next letter. Go up here. These all have 2 little r's. This one does, but there's no capital Y, so that can't be it. Capital Y, capital Y, capital Y, which means all of these will have the same phenotype. So I'm going to put a 3 here down at the bottom. And my last one, wrinkled and green seed. Well, I know wrinkled is 2 little r's and green is 2 little y's. So I'm looking for this combination, 2 little r's, 2 little y's. And you can see in this cross, there's only 1. So now I'm going to write my ratio over here. It's going to be 9 to 3 to 3 to 1. 9 to 3 to 3 to 1. Whenever you cross two heterozygotes with a dihybrid cross, it's always a 9 to 3 to 3 to 1. I will have several practice problems for you guys. All right, I will have a problem that you can try, and I'll also have an assignment for this, but we're not done quite yet. Dihybrid cross practice. This is what the homework will be based off of. And I'll have a digital version of this. I'm just using this to show you how we would go about answering the questions that'll be on tomorrow's homework assignment. Or today if you're watching this video on Thursday. Okay, I have two parents here. Here they are. What I need to do is I need to put those gametes for those parents across the boxes here. Now, when I made this, I only have 4 options. This is for one parent. So I'm going to write one parent gamete across the top and the other one across the bottom. And then I'm going to use the Punnett square on the larger Punnett square to do this. So I'm going to use my FOIL method, which first is going to be big G little y. Outside, big G little y. Now, those are the same. Those are the same because the second trait is homozygous recessive and there's only one combination that it can give. Next one, inside, big G little y, outside, big G little y. It's going to be the same difference because this parent does not have any capital Y's. Okay, so that is my first parent. My second parent, first, big G, capital Y, outside, big G, little y, inside, big G, capital Y, outside, big G, little y. Well, again, I have only 2 combinations because one of the traits on my parent is homozygous, which means a capital G is missing. I'm going to go back to my Punnett square here and fill it in. This is the part of the video where I want you to pause and I want you to fill in this Punnett square on your own. And after you've done it, I want you to play the video again and check yourself and see how you did with filling this in. Hopefully you have filled in the Punnett square on your own, and then I want you to check it using this key, just see how you did. If you use the 2 different colors, it's easier to see and recognize, what's what. Okay, so what we're going to do now is we're going to do a phenotypic ratio for this cross. And I'm going to look at the other questions that are on the assignment that is a sample of what you'll see. So first we need to write what are the 4 different phenotype combinations that we can see. Well, first, what are we even dealing with? We're dealing with a big G. Well, I know big G stands for green pea pod. So it's a green pea pod and a yellow pea pod. And I know Y is for yellow seed. So I'm going to have both my dominant traits. So my first one is a green pea pod with a yellow seed. Both of those are dominant traits. So I'm going to have a big G. I'm looking for a big G and a big Y. And I don't care what the second one is, so as long as I have one, I have what I need. Next, green pea pod. And instead of yellow, we have a green seed this time. Green pea pod is a capital G, but green seed is recessive, so it's 2 lowercase y's. So that's what I'll be looking for on that one. Our next one here, instead of a green pea pod, we now have a yellow pea pod. So here's my recessive, and then I need the dominant seed color, which means I'll be looking for 2 G's and 1 capital Y with whatever's with it. And my last, I need a green pea pod which is recessive, with a green seed, which is also recessive. So I'll be looking for 2 little g's and 2 little y's. Now we're going to go up here. We're going to find in all of our types here, we're going to do green pea pod first. Green pea pod, we're looking for big G, big Y. Okay, here's one. Nope. There's one. There's one. Nope. And none of the rest of them are. Okay, I'm going to count them up. So I have 4. Next, green pea pod, green seed. So big G with little y's, big G with little y's, big G, little y's, and this one. None of these have big G's. So again, we have 4. Next is a yellow pea pod and yellow seed. So I'm looking for 2 little g's and a big Y. Here it is. You might notice a pattern by now. Again, we have 4. And a green pea pod and green seed. 2 little g's, 2 little y's. 1, 2, 3, and 4. All right, so here's the information. We're going to come back to this. I'm going to go back to the questions here. So I filled in my Punnett square. Let's look at the bottom. List all of the possible genotypes in the offspring. So when you do genotypes, I'm going to show you the easiest way to list all of them. There are 9 possible genotypes for any kind of trait. That's all you can see. So there's a easy way. So I want to write 3 sets of big G's. And these big G's are all going to go with, it can go with a homodominant, a hetero, or a homorecessive. Well, now I have big G, little g, and all of these can go with the same homodominant, hetero, homorecessive. And my last set is my homorecessive, which again can go with homodominant, hetero, or homorecessive. Now, not all of these are actually possible in my cross above. And I'm going to show you how you figure that out. So on my cross here, look at my parents. I can get a big G from the top parent, but there are no big G's from the bottom parent, which means none of these are possible. You cannot get 2 big G's because these only give little g's. You can get the heterozygotes and the recessives. All right, so now we need to check, well, what about my Y's? Big Y is not possible. This parent does not have any big Y's in it. So I can cross that and that out. So there are only 4 possible phenotypes, which are those, based on the parents and the cross. Calculate the genotypic ratio, calculate the phenotypic ratio. You will only do this when it's certain ones, but this is one that we can do. Our phenotypic ratio we already have. Our phenotypic ratio is this. We have all 4s, which means it's 4 to 4 to 4 to 4. When you have a nice setup for a phenotypic ratio that's even, your genotypic ratio is identical to it. That's also 4 to 4 to 4 to 4, because you only have 4 genotypes and 4 different phenotypes. This one, list all the possible phenotypes of the offspring. We already did that. I did it over here. So again, that's our green pea pod with yellow seed, green pea pod with green seed, yellow pea pod with yellow seed, and green pea pod with green seed. So those are the 4 combinations. Now let's do the very bottom of this, and then we're finished with this problem. Probability. Probability has to do with the percentage of offspring. So how many will have this? The way you figure this out, this ratio lines up with all of these problems. So I'm going to start with putting 4 for each one. Well, this is 4 out of something to do a probability. Well, it's 4 out of 16, and I know this because there's 16 boxes. So 4 divided by 16, divided by 16, 16, 16. 4 divided by 16 is 1 over 4, and if I put that in a percentage, it's 25%. So the probability of getting an offspring for any one of these traits is 25%. So this is the information that you'll need to answer the problems, the homework. It's based off of this. It's the same thing. I'll just have different parents. Again, please make sure you watch this video. If you have any questions, let me know. And have a wonderful day. Bye.